First of all, it's a great pleasure and an honor for me to be commemorating Rita Levi-Montalcini. I mean, she is a giant, and a woman, doing what she did at the time that she did it is absolutely magnificent. So she's always been an inspiration. I was fortunate to be able to have met her, and so I think it's very befitting to come back and honor her. So I start. I think everybody's seen this picture. I think this is an early picture, and I think she was inspiring. I mean, just an amazing woman doing what she did when she did it. And I think she's also influenced essentially that now we're getting a little bit more parity. Not perfect, but we're getting closer, at least, to parity, and that should be our goal. I'm going to show you another slide, which is sort of a family tree, but unusual in a sense that it's not genetic. But I think what it illustrates is the importance of mentorship. At the top is Giuseppe Levi, and he had three remarkable students: Salvador Loria, Renato Del Becco, and Rita Levi-Montalcini. All three of them became Nobel Prize winners. Just remarkable. I think you recognize those. Luria had Watson when he was at the University of Indiana. Del Becco had David Baltimore. And then Watson had Mario Capecchi. So it illustrates this, to get such a combination is certainly not random. And it illustrates the importance of having somebody that you can see has succeeded and saying, if they can do it, why not I? I think it gives you a capability to dream and then fulfill your dreams. So I'm going to start by talking about a neuropsychiatric disorder in a mouse. And I think most of you would think a mouse has no behavioral similarity to humans. But I think I'll try to see whether we can get a connection. We started in this by essentially serendipity. We were studying a set of genes called Hox genes. I think many of you may be familiar with Hox genes. They're important for establishing the body plan. It makes sure that our head is here, our arms at this position. It sort of gives you a coordinate system for the entire body, and also subcoordinate system, for example, in the limb. Again, Hox genes are important for both distal, center to distal axis, as well as anterior to posterior axis of the body. And we've knocked out all of these genes and then started asking, what are they doing? And I should always say we is the royal we. It's always graduate students and postdocs. I simply help and encourage. And the particular gene that Joy Greer was interested in is HOXB8. And the nice thing about Hox genes is that their position along the chromosome also tells you where along the body axis they're functioning. So genes at this end of the complex are working at the head end, and genes at this end of the complex are working at the tail end of your body, okay, and everything in between. So Joy Greer, one of my graduate students, her project, among many other projects, was to disrupt HOXB8 and ask what happens. And she looked at it, and it looked pretty normal, which was quite unusual because in many of these genes, even having just one defective copy was enough to give you a visible change, a phenotype. Okay? So she didn't see anything wrong with it, so she decided to look at behavior. And because mice behavior is mostly at nighttime, she used an infrared camera and then simply took movies over a 24-hour period, and then asked, what is a mouse doing over that whole entire period? And looked at every possible behavioral deficit. Okay? And And the only thing that she saw different was actually grooming. Okay? grooming is important and stereotypic, and all organisms are groomed. C. elegans grooms, a little worm, Drosophila groom, mammals groom, all mammals groom, and we groom. Okay? And grooming is important because it's a way of shedding, getting rid of things like bacteria and viruses in your body, and it has an enormous effect essentially on longevity of people. Okay? And the syntax is shown here. So you start, you go into the shower, you take soap, lather it, and start working on your head, and then you work down your body. And that's what all animals do, and that's shown here. It's simply they start, they lather their paws, and then start working on their head, and then work down their body. And the syntax in this mutant mouse is not affected. The only thing that's affected is how many bouts per 24-hour period, as well as how long those bouts last. So they excessively groom. Here's an example of some of her data. So the black bar is a mutant that has 2 defective copies of HOXB8, and gray is a sibling that is also matched for sex. So what you're looking here is how much time have they spent eating, and what you can see is that they average out spending the same amount of time eating over a 24-hour period. And she looked at many, many, many different behaviors, and the only one that was different was how much time they spent grooming. And that's shown here, and what you can see is that a mutant spends about twice as much time grooming as normal. You can also induce grooming. You can actually spray a mouse with water, and that induces it to groom. Okay? And in this particular case, we actually used wild-type mice from the field. We wanted to make sure the genetic background wasn't affecting it, so we crossed our mutation to a wild-type mouse, a real wild-type mouse, and yet, if you follow the genotype, then you have twice as much time grooming. Okay? Now, I told you one slight error, and that is if you spend over a 24-hour period doing too much of one thing, you have to take it at the expense of something else. And what these mice do is spend less time sleeping. Okay? So more time grooming, less time sleeping, and it's pathological, and I'll show you that, just a short movie. And what you'll see is the mouse grooms and grooms and grooms, and then when it moves to the side, what you'll see is that it actually removes the body hair in the process of grooming. Okay, this mouse is going to turn, and there it's removed all the body hair. And it continues to do that until it has lacerations, and then we put it to sleep. Yeah? So this is very similar to OCD. For example, obsessive compulsive disorder. Humans wash their hands and continue to wash their hands until they have lacerations. And then at that point, they're still not getting a signal saying your hands are clean, go on and do something else. And that's what makes it pathological. Now this is another sort of unexpected result. If you put a normal mouse in the same cage with a mutant mouse, it'll groom it. Okay? But the pattern is completely different. Normally, a mouse removes all the body hair, what we call the ventral side, the chest side. And here it's removing it from the back of the other mouse, because that's what's exposed to it. But this gave us a very important clue, and that is it's not likely to be an itch. That is, it's not likely to feel an itch on another mouse, okay? And that told us that it's likely to be something to do with the central nervous system, in which actually grooming behavior is actually been shown simply by doing very crudely removing different parts of the brain, then you can disrupt grooming behavior. So that told us we had to concentrate on the brain. Okay, so then we switched. Now this is Jacques Juan, another talented student in the lab, a graduate student. And taking movies is very robust. And you can get the data, but it takes an enormous amount of time because she's looking, Joy used to look, frame by frame, what is this mouse doing over a 24-hour period? Huge amount of work to get the data. So now what we have are platforms that are extremely sensitive to vibration. Okay, so no matter what your motor output, you have a different vibration. If you're walking, it's one vibration pattern. If you're drinking, it's a different vibration pattern. If you're scratching, different vibration pattern. Grooming is a different vibration pattern. So the platform automatically reads out the whole behavior over 24 hours, and nobody has to be in the room. So the handler is not affecting the behavioral output. Okay? So we're simply getting a lot of data. And again, what you can see now, this is relative about 50 mice. Again, mutants spend twice as much time grooming as normal. Okay. So we looked in the brain for the expression of HOXB8, and we couldn't find it. Okay? And that was sort of, we could see possibly a little bit. It looked sort of like, what we call salt and pepper pattern. But it wasn't focused, and we couldn't pinpoint which cells were actually responsible for the behavior. So under these circumstances, what you do is simply make it a marker. That is what we have. We isolate a gene from a jellyfish that makes a product that fluoresces green. You stick that in the middle of another gene, and then wherever that gene is functional, then you get green fluorescence. Okay? So then that gives you a great readout. So here in the experiment, we have essentially a locus. It's a Rosa locus, which is ubiquitously expressed. This is our GFP protein. We flank that with LOXP sites, and then there's a protein that essentially excises, in the presence of that protein, excises it does a recombination event between these two LOXP sites, excises a stop sign, and then gives you a readout wherever Cre is produced, and Cre is produced in only cells that express HOXB8. Okay? So by doing that cross, we specifically permanently label every cell that's ever expressed HOXB8, and then they stay green. Okay? So that gives us a lineage way of tagging this particular, all the cells in the body that are expressing a green cell, and therefore in the brain. And this gave us an enormous surprise. The only green cells in the brain were microglia. Microglia are immune cells. They're made in your blood system, and then are used for infection, for example. And then here you have all of us. What we're saying is that microglia are responsible, and a defective microglia are responsible for behavior. Okay? Because here is the green cells. Here's a pan-neuronal marker microglial marker, HOXCD11B. And then here's yellow cells, which are both green and red, saying that it is a microglia. But also note that not all microglia are red, okay? Are green. So in this case, okay, so it's about 30% of the cells are actually labeled with our HOXB8 lineage tracer. Okay? So it has to be at least two types of microglia in the brain, ones that expressed at some time our gene, and ones that didn't. Okay, so what I've told you just now is that microglia are responsible, for the defect. Okay? So that was quite unusual. That is, how could microglia be controlling neural circuits? So the next question we ask is simply, where is HOXB8 expressed in the immune system? And here we simply took blood, and you can separate blood according to different markers, distinguishing myeloid from lymphoid cells, B cells, and T cells. And what we're showing you is the data in the absence of Cre and in the presence of Cre, and what you can see is that total population is shifted over in the presence of Cre, becoming now green. Okay? So this tells us it's either progenitor cells capable of making both lymphoid and myeloid cells, or the hemopoietic stem cells themselves. Consistent with being in stem cells is simply if we take blood and separate it according to two common stem cell markers, c-KIT and SCA1. Here's a double positive. That's the stem cell population. Again, in the absence of Cre, in the presence of Cre, we shift that whole population over. Okay, so that tells us indeed it is in the stem cell compartment. Okay? So this made a very strong prediction. If it's in the blood system, we should be able to cure the psychological pathology with a bone marrow transplant. Okay? simply lethally irradiate the mouse and then supply them with normal bone marrow, and we should cure the mouse of pathological overgrooming and removal of body hair. That's the prediction. So again, Shaquan, I asked him to do that. He said, do a bone marrow transplant in a mouse. He said yes, and so he proceeded and did it, and it worked. First of all, do we get green cells in the brain by doing a bone marrow transplant? And the answer is yes. It takes a while. It's the same story in humans. It takes about 12 weeks, essentially, for a bone marrow transplant to work in humans. But at that, then we get very good transfer, again, after lethally radiating. Okay, and lethal radiation does two things. One is it destroys the bone marrow that's there, and therefore now you have to add new bone marrow in order even for the mouse to survive. And the other is that it breaks the blood-brain barrier, so cells from the outside can get into the brain, okay? And that says the experiment does work because we introduce these cells into the blood system. Okay, and here's the actual experiment. So here is a mutant mouse about 3 weeks after bone marrow transplant, okay? And what you can see is the mouse has removed all the body hair. If I showed you this a few weeks later, what you would see is lacerations all over the body and then a lot of bleeding. But after the bone marrow transplant, now 12 weeks after bone marrow transplant, all the lacerations are healed, all the hair is grown back, and then if we look at how much time they're spent grooming, here's the wild type, here's the mutant, and now the rescued by the bone marrow transplant goes back normal. So now all of that behavior is permanently cured by a bone marrow transplant. Okay? So that prediction held. We can do the opposite. We can put mutant bone marrow in a normal mouse and give it the pathology. So that says it's not only necessary but sufficient for the giving rise to the pathology. Okay. So there is a difficulty, and that is Hox genes do a lot of things. They normally have 200, 300, 400, 500 different target genes, and they're doing many, many things to the mouse. And one of the things that HOXB8 is also responsible for is making the interneurons that receive inputs from the periphery in terms of pain. Okay? So these mice are insensitive to pain. And if we look at where the gene is expressed and where it affects, it's the dorsal aspect of the spinal cord. So here we're looking at a marker that is the neurons that are coming in, the sensory neurons that are coming in. Here, and these markers actually mark the interneurons that receive those inputs. And here's the normal pattern. And here in the mutant, what you can see is that you're making a lot fewer of these interneurons, and they're also much more dispersed. Okay, so that's the phenotype. And then you can also look at the behavior. And what you can do is just put a mouse on a hot plate and ask how quickly does it react to the heat. And you can measure that. And what you can see is the mutant mice spend about 3 times longer responding to high temperature. Okay. So the first clue that we could actually separate out those two phenotypes was actually in the bone marrow transplant itself. Okay, I told you that bone marrow transplant completely cures the mouse of the overgrooming and hair removal. However, it does not appear to affect insensitivity to pain. Here's the wild type, here's the mutant, here's the bone marrow transplant, and it's pretty much the same as mutant. Okay, it looks like there's maybe a little bit of correction, but not significant. Okay, so the way to really study this is simply to use conditional mutagenesis to separate out those two phenotypes. We can inactivate it in the bone marrow and ask what happens. We can inactivate it in the spinal cord and ask the consequences. Okay? So we use Ty2-Cre because that's expressed in hematopoietic stem cells. So we can inactivate it there without affecting the spinal cord and see what happens. First of all, does Ty2-Cre actually label those microglia? And that is yes, they do. That's good. And here's the actual experiment. So here we are just inactivating in the bone marrow. Then we actually recapitulate removal of body hair. We recapitulate how much time spent grooming, but we do not affect insensitivity to pain. Okay? So that says we've separated it from that direction. We can do the opposite. We can inactivate it in the bone, in the spinal cord, and specifically, and then using conditional mutagenesis, and then ask the consequences of that. But here it simply shows you also the phenotype in the spinal cord, and what you can see is that it's not affected. It's still wild-type pattern instead of mutant. So this is mutant. Here's type 2 Cree, and that's similar to wild-type in terms of how many interneurons there are and how tight it is with respect to their lamination in the spinal cord. Okay, so that is that experiment. So now we can do the opposite. We inactivate it specifically in the spinal cord and then ask what happens. And for that, we used a parallel of HOXB8, which is a partner on another chromosome, and that has the identical expression pattern in the spinal cord compared to HOXB8. And knock it out and ask what happens. And what you can see is no hair has been removed. There's no effect on how much time they spend grooming, but now we completely recapitulate the insensitivity to pain. Okay, so that allows us to say, indeed, HOXB8 is responsible for those two functions, but they're completely separate. They're not overlapping with respect to how they're doing that. Okay? And again, if we look at the spinal cord, now we see the pattern is identical to the mutant pattern instead of having a nice laminar structure compared to wild type of those particular interneurons. Okay. So now we made a mouse where we can label those two different populations of microglia and study what they are. And the reason this is important is because in the literature, they say there's only a single source of microglia, and all of those cells come from the yolk sac in what's called primitive hematopoiesis before actually definitive hematopoiesis. So what we did is label all microglia green, and then the ones we're interested in, we label red, and then what you can see is the red ones are yellow if we look at both. So this is a coincidence, and again, about a third of all microglia are HOXB8 microglia. Okay, so with this configuration, we can follow these cells in the same mouse. And this is actually live imaging using two-photon microscopy in a live mouse looking at the brain directly from the outside. Okay, so the first question we ask is, where do our cells come from? Okay, and the reason, again, just to recapitulate, is that it's a very nice literature showing you that all microglia essentially are derived from yolk sac, and we're saying, there has to be something different because we have two populations that appear to be doing different things. So the first thing we asked is, when do yellow microglia actually get into the brain? Green microglia, the ones from the yolk sac, get into the brain at 9.5 days of gestation. A mouse has 19 days of gestation, and at 9.5 days, the cells are born in the yolk sac and directly migrate into the brain. And that's the green population. The yellow population, we don't see any cells, yellow cells in the brain until 12.5 days of gestation. So they're coming in, presumably, at a much later time. There's actually two possible hypotheses here. Okay? One is that there's a new source of yellow ones coming in later. The other is that a subgroup of green ones turn on HOXB8 in the brain and now become yellow, because those are the two possibilities according to this experiment. Okay? Now, the prediction is then, if they actually come into turned on in the brain later, then we should see that. There should be transcripts in the brain at a later time. Okay? And we looked at transcription of this gene when the actual gene is expressed, and what we find is that the expression is actually in the yolk sac. So it starts in the yolk sac, okay? But then the expression drops way down. This scale is broken here and it goes way up here, but simply so you could see the background expression. But this is the brain, okay? We're looking at the brain all along, and there's zero, actually, less than a couple of orders less than one transport per cell. And we cannot detect any expression of our gene in the brain. So that tells us that the source of the second population has to come from the outside. There's no expression inside the brain. Okay? So we can actually sort it and actually see what it looks like. And what you can see is that this population is essentially showing up in essentially AGM as well as in the fetal liver. And there's very little, about 3% is present in the yolk sac. So there, and what happens also, if you actually look at the numerology, it's not only that the cells, instead of going to the brain, now go to the AGM and then fetal liver, but also that this particular population is uniquely expanded in those latter tissues. Okay, and that's shown here. So here we're looking how much of these cells, yellow cells or red cells, are present in the yolk sac, in the AGM, as well as in fetal liver. And you can see that at the end, 100% of the cell population is actually in the AGM and the fetal liver. So the data looks like this population of cells is born in the yolk sac, but then seeds the AGM, which then seeds the fetal liver, and in the process expanded, and then goes to the brain. So it has a very different trajectory, and because of that, now, because of being in the new environment, it can now pick up new properties that differentiate it from the microglia that are already in the brain coming directly from the fetal liver. Okay. There is a gene that distinguishes primitive hemopoiesis from definitive hemopoiesis, and that's CMIB, so we can inactivate it and ask what happens to the green population and red population. It's very easy to see the mutants because they don't have any blood or red blood cells, and therefore you don't see the vasculature in the embryo as normal. So these are wild type. Here is using actually CRISPR-Cas9 to inactivate this gene in the embryo, and then what you can see is look at these populations, and then what you can see is this the progenitor population, which is up here, which is c-KIT high and also red. That population is almost wiped out essentially by CMIB. And quantitation is shown here in terms of how many cells get into the brain. Here are yellow cells in a wild type mouse and then in a CMIB mutant, whereas it does not affect green cells getting into the brain, which is MIB independent. Again, arguing that this is a separate population of blood cells. Okay. So how similar and dissimilar are these two populations? So we can ask that because we can follow these cells. First of all, we can simply ask by looking at all the genes that are expressed in these particular cells and comparing each other. And there are only 21 genes that are distinguishable from the green population. So out of 30,000, only 21 genes are a few-fold overexpressed in this population. So these two populations are extremely similar, even though they have a different route to getting to the brain. Okay, that's what that says. We can also ask, what do they do? One of the things that microglia do if you have damage is they actually send their pseudopods. Microglia almost look like neurons. I mean, they have a cell body and all of these extensions. Okay, and so what we're going to do is in a live mouse, we'll do damage right here with using a laser coming from the outside. We'll kill a few cells here, and then all the microglia around that damage will extend all of these extensions right to where the damage is. Okay, so here are some green ones, and here are some yellow ones. These are the HOXP8 ones. These are the ones coming from the yolk sac, and the damage will be right there. And you can watch that. Zap, and there's the damage. And then what you can see is they extend their pseudopods right to them, and then are going to then correct that damage. Okay, and in this assay, those two populations are indistinguishable. And this is one of the main things that microglia are doing, is fixing damage, for example, from a stroke or an infection. Okay. The other thing we can do is to cut a nerve on the outside and where the cell bodies are inside. These are the facial nerves. Okay. Remember, we can't stick a needle into the brain because then the microglia would just go to the needle. Okay, so we don't want to be causing damage inside the brain. We want to cause it from the outside. And what happens if you cut this nerve, the cell bodies are in the brain and then die because now they no longer have connections. And then all the microglia go to this damage. And what you can see is we've done essentially cut on this side of the brain, and then this is the other side of the brain of the same mouse where we haven't cut the nerve. And here you can see all the microglia going to that place, and also yellow ones. And again, there are, in this particular part of the brain, there are not as many yellow ones as green ones, but if you simply measure how well they function, the yellow ones perform a little better than the green ones, but it's pretty much similar. So again, by that assay, again, the yellow population and the green population seem to be quite similar. There's another thing that microglia do, and that's you always produce about twice as many neurons as you need. Okay? And then there's a process that goes on during embryogenesis and as well as actually when you're further developed after birth, and where you eliminate those neurons that have weak connections and thereby strengthen the ones that are stronger. And microglia are intimately involved in this process, and they can recognize in an activity-dependent manner which are the strong connections and which are the weak ones, and eliminate the weak ones. Okay, and that's synaptic pruning. And we can do that, again, by putting a dye in the eye. Okay, so we put the dye right in the eye, and then it travels and goes back to the thalamus right here, and then there it labels the axons that are present, and then we can ask whether the microglia in that vicinity are actually pruning, because then the synaptic material will be inside the cells. Okay? So that allows us essentially to assay how well they are with respect to pruning, and you can also show that it's activity dependent. So here is essentially the retrograde labeling, and then here is the microglia itself. And I don't know if you can see it, there's little blobs there, and that's actually the activity of the label that we introduced into the eye. And here is a yellow one, and here's a green one. And if you quantitate how many there are per cell, and you can actually rotate the whole cell and then show that the label is inside the cell, not just stuck to the cell, then under this assay, again, those two populations are equally effective in terms of synaptic pruning. Now, where do they differ? And where they differ is where these microglia are in the brain. Okay, so here's a sagittal section of the brain, and this is all input coming from the spinal cord. Okay, because the spinal cord, remember, those inner neurons are labeled, and so their extensions into the brain are also read. But if we look out here, what we'll see is individual microglia, and whether they're green or red. Okay, whereas the HOXB8 ones are red, and the non-HOXB8 ones are green. And then you can simply count them and ask, here's the primary and secondary motor cortex, here's the anterior aspect of the striatum, orbital cortex is right here. And then if you look at these different regions, what you can see is that they vary in where there's high HOXB8 ones relative to green ones, okay? And this region right here is very interesting because by functional MRI, this has been defined essentially as the OCD circuit, okay? Patients with OCD, this area is highly active, whereas if compared to controls, and then other regions are not affected. And where we find HOXB8 microglia are actually in the same region as been previously defined in humans as the OCD circuit. This may be a coincidence, but if it is, it's a very interesting coincidence. Okay. So the other thing I want to remind you is that the HOXB8 microglia are about 30%. Okay, so they're a minority pool, and yet 70% of the cells can't correct that defect in HOXB8, or else we wouldn't see any behavior. Okay, so functionally, these two populations have to be doing something different. Okay. Now, the other aspect is, I don't believe that what we're looking at has to do with OCD. And why do I believe that? It simply looks like it's a very elaborate system to simply be monitoring one particular circuit. And the question is, what are we really looking at? And one possibility, this is pure speculation, is anxiety. Okay? And the reason for thinking this is that two things. One of the things that people have been doing now gene association studies for many, many different neuropsychiatric disorders. Okay? And two things can come out, which is also perplexing. One is the same set of genes no matter what neuropsychiatric disorder they're looking at. Okay? So somehow mechanism and output is not correlating with each other. The other is that about half the genes they pull out, no matter which neuropsychiatric disorder they're looking at, have to do with the immune system, which is also perplexing. Okay? So in tying this together, anxiety is also associated with many, many different neuropsychiatric disorders. Okay? If you look at OCD, and particularly trichotillomania, which is removal of the body hair, what you find is all of them say, and I've interviewed hundreds, they say when we're anxious, we go into the pathological behavior. When we're not anxious, we can control that pathology. Again, indicating that there may be an association between anxiety and that output. And this is true not only for OCD, but also for other neuropsychiatric disorders. So one possibility is that this system is actually controlling anxiety. So the first question we can ask is, are mice anxious? So there are a number of assays used for anxiety. One is what's called an elevated maze. Okay, you have a maze up in the air, and then one set of arms, the closed ones, okay, have walls on either side, and then the other is completely open. So they, on this maze, on the open side, they can look way down, and that's, for a mouse, that's a long distance, okay? And that an anxious mouse will spend a normal mouse will spend equal amount of time on either branch. If a HOXB8 mouse stays all the time in the closed quadrant, okay, and it doesn't venture out into the open. That's one measure of anxiety, and by this assay, these mice are very anxious. Another measure which is similar is what's called an open field. You have a big open field and again sides of the field, okay? And then you put the mouse in and ask where are they spending their time, and then this traces their movement. And what you can see is a HOXB8 mutant spends less time on the inside compared to the outside, okay? So again, this is a separate measure of anxiety, and again, these guys are very anxious. And then another very simple assay is to simply have two boxes, one with a light in it and one in dark. Okay, a normal mouse will explore both boxes, and an anxious mouse will go into the dark one and stay there. And again, if you measure this, what you see is the mutant mouse spends a lot less time in the light compared to normal. Okay, so by all of these criteria, these mice are very anxious. Okay, the other thing is that about 25 to 50% of patients with OCD and trichotillomania, which is the removal of body hair, react to certain drugs, and particularly SSRIs, okay? So what we can see, that fluoxetine now, essentially, if you give it such a dose that it doesn't really affect the wild type, but it decreases how much time they're spent grooming of a mutant mouse, okay? So essentially, it makes it more normal. Okay, so the same drugs that are effective in humans are also happening to work in these mice. Similarly, you can do the same thing in the open maze, and then you can do the same thing in the dark, and again, get it back to normal. So you can correct essentially the behavior with a SSRI, a serotonin uptake blocker. Okay, so in closing, here's a circuit that's been defined essentially as OCD by functional MRI. And what we're seeing is microglia are somehow modulating that circuit. We have no idea how that modulation occurs. It could be chemical. Microglia produce cytokines, they produce chemokines, they even produce neurotransmitters, glutamate, for example. So they could be producing compounds that can modulate that circuit. But there's other more interesting possibilities. Waki et al. In Japan has shown that if you actually watch microglia in vivo in a live animal, they're always extending these projections all the time. They're waving around in space, and then they project them, retract them, and so on. And you actually can calculate that in about an hour's time, because there are lots of microglia, they can cover the entire parenchymal surface. So essentially, they can sense the entire surface and what's going on. But then all of a sudden, what you see is they stop, and then they essentially engulf or surround essentially a synapse completely. And you can actually, by EM, see that that membrane is right around the synapse. Okay, it's an enormous amount of work, and again, it's activity dependent. Okay, so you can show, for example, very crudely, for example, if you cover one eye, then that eye, that doesn't occur wherever that particular, those exons project, okay? So it's activity dependent, and it's an enormous amount of energy, and what is it doing, and can it be modulating activity across that synapse by that function? So those are things that now we can do simply because there are tools to be able to do that, and that is optogenetics. So we can start asking, do microglia directly talk to neurons, and particular neurons we know where to look, and then if they do talk, what is the language, and how do they modulate neural circuits? So that's where we're going in the future, and I thank you for your patience, and I'd be happy to address any questions. Thank you.
Thank you very much indeed, Professor Capecchi, for this very inspiring and exciting lecture, showing us recently. I'm sure that many people in the audience are excited about your data. Questions, whilst people warm up, I will ask myself one question.
Sure.
Of course, you showed us in a very, very, thank you, in a very nice way, how other cells other than neurons can modulate behavior, and this is a very, very important concept. I was wondering, are you thinking that might it be worthwhile looking epidemiologically at the millions of people who have had bone marrow transplants, if there are behavioral changes in one direction or another that could be traced possibly to the bone marrow transplant?
Good. Now, excellent question. I was estimating now probably 700,000 bone marrow transplants worldwide have been done. Okay, so there's a lot of data. All of these neuropsychiatric disorders are roughly in the 1% to 3%. I mean, they're very common. It's not rare. So if you've done that many bone marrow transplants, there should be a lot of overlap. So I actually have been calling all of the different bone marrow transplants in the United States and asking, have you experienced anything unexpected after a bone marrow transplant? And remember, it should go both ways. Okay, we've shown that if you put good bone marrow into a mutant, then you correct it. And if you put bad bone marrow into a normal, you give it the pathology. Okay, I've called all of these centers and they say, no, we haven't, we aren't keeping track. However, I have a pile of emails about this high from patients, okay, that say very strange things have happened to me. Okay, and again, in both directions after a bone marrow transplant. Unfortunately, that's not data, okay, that's a selective pool. But I think it's something that one should address. I mean, I think it should be interesting. The other thing that you can ask is, does this have anything to do with humans? I mean, we know what gene we knocked out. 3% of the people have trichotillomania. In any population, people worldwide, I've looked at 8 different populations worldwide, always about 2.5 to 3%. So we can get DNA, and so we can ask, do they have a defect in this gene? Okay, and there are 2 things that are important. One is we're looking at homozygous mutants, okay? And fortunately, unless you marry a cousin, you're not likely to be homozygous for a recessive mutation, and that's a good thing, okay? However, what we're saying is, can you have a defect in this gene plus something else? What is that something else? It would be a gene also in the immune system. When coupled together, would be equivalent to then being homozygous for HOXB8. Okay? So we went to the population, we looked at their DNA, we know what gene to look at, and there is a polymorphism actually that's quite common. About 25% of the people have it, and it does track with the mutation. Unfortunately, we can't do the experiments in humans. Okay, so what we've done is take that polymorphism and put it into our mouse, and now we're in the process of essentially asking does it now have pathology? Okay, and particularly does it have a grooming pathology? So I think we can, there's a hint already, because there is a polymorphism that's present there. We would predict that it will result in loss of function in terms of loss of transcription, or at least of that gene, and it's consistent with being a splicing defect. So I think we may be able to make a connection with the humans, and we should have that data, and then that would allow us essentially to close the circle. So I think there are a number of interesting things that we can do with humans as well as with mice. Yes?
Mario, that was wonderful. How is the distribution of the microglia in the brain regions? Is it homogeneous? I would expect it's not.
Yeah, no, it isn't.
I mean, I know that the difference of the two type of, but the OXB, is it homogeneous in the various brain regions or not?
Yeah, no, it's distributed in the same regions that previously people identified as being what they call, I mean, it's a fairly large circuit because, it involves essentially the striatum, the motor cortex, and the orbital cortex, okay? So it's a fairly large region, but nonetheless, that's where the HOXB8 ones are at much higher level compared to other regions of the brain. And that's reproducible from mouse to mouse. However, so far we haven't defined how these cells know where to go. And that could be either active or passive. And active meaning that certain neurons are giving out some kind of a signal that says come to this region, and then the microglia would have to have the appropriate receptors. And passive may be that by going into the brain by a different passage, at a different time during development, they would actually be in different regions. So, and we're trying to work that out also.
Other questions? Well, of course, Mario Capecchi will be around, so I'm sure that many of the younger scientists will want to talk to him. And so we now have coffee break. I would like to thank you very much for this exciting lecture, and the coffee break. And please reconvene here at 11:30